Hello, my name is Dr. Sarkisian. This is Biology 430, Neuroscience, and today's lecture is going to focus around the etiology of action potentials. Thus far, we talked about some of the basics of all action potentials have very characteristic profile, which are represented here on this slide. The resting potential, as we have discussed already, of a neuron is somewhere around here, which is minus 65 millivolts. Now, when an action potential starts developing, we see a rapid depolarization of the membrane, which means the charge that was inside, the negative charge that was inside the membrane rapidly becomes positive. So this phase in which this rapid depolarization occurs is called rising phase. Rising phase ends with what's called an overshoot, in which the membrane potential actually becomes positive. That means the inside of the cell becomes more positive than the outside. Following this overshoot, we have the third phase, which is the falling phase, where the potential starts basically normalizing, which for our neuron is that the inside of the cell is more negative than the outside of the cell. However, there is a brief period in which the potential becomes actually less than the resting membrane potential, which is known as undershoot. And this is a very important property of a neuron. Now, we will talk about it later, but after the undershoot, the membrane potential slowly comes to its resting state. So the whole action potential, if we look at from 0 to, let's say, about 2.5 milliseconds, that's how long an action potential, a single action potential, lasts. And this is what it looks like with the distinct phases: the resting phase, rising phase, overshoot, fall, and undershoot. And then it reverts back to a resting potential. Here is a summary slide for your reference for later. This outlines the rise, the overshoot, fall, and the undershoot properties of an action potential. So to go back and look at our initial example of a thumbtack being piercing through skin and generating pain and a reflex, basically this perception of pain is caused by generation of action potential in very particular nerve fibers in the skin. Now, the membrane of these particular types of fibers are actually gated sodium channels. Gated means that they respond to a particular stimulus. They can, in this particular case, these sodium channels open up in response to mechanical stimulus, such as stretching. So, what is the... We will talk about these in greater detail in chapter 12, but let's look at it. Let's look at an overview of what's happening here. The sequence of events is as follows: a thumbtack enters the skin. The membrane of these nerves is stretched. This stretching opens up sodium channels, and given that the sodium concentration outside of the cell is greater than inside, the sodium starts passively diffusing into the cell. And this diffusion into the cell, of course, carries sodium's positive charge with it and results in depolarization of the membrane. So, as a result, the cytoplasmic side, the inside of the cell, becomes increasingly more and more positive, and after a certain threshold, it generates our action potential. It is very important to recognize that an action potential is an all or nothing event. The depolarization of the membrane must reach a particular threshold for an action potential to be triggered. So unless the membrane potential reaches a particular magnitude, it will not generate an action potential. The depolarization will basically shut down and the cell will go back into its resting state. But if an action potential is generated, then we have a chain reaction that one generates another, and that propagation becomes more or less perpetual for that particular event. So that opening of ion channels, changes in resting potential or the membrane potential, all of them could be certain stimuli for certain other channels to be activated or deactivated. And we will talk about this in depth shortly. So, how did we come to understanding all of this? Well, obviously it took quite a few years and quite a bit of money and research. But it all boils down to a single very interesting neuron, or an axon, I should say, that comes from a giant squid. It's its main nerve fiber that spans throughout the entire body. And initially, when it was discovered, scientists actually thought that that was part of the vascular system, a blood vessel. It was too thick, it was too big. But lo and behold, it was a nerve fiber. So having such a big nerve fiber opened up quite a few opportunities to do some really interesting research. So based on that and many other research, we now know that action potentials could be as infrequent as 1 hertz, which is one spike per second, or it could be multiple, up to 50 hertz, which is 50 spikes per second. But the maximum firing frequency could be about, well, has been measured to be about 1000 hertz, 1 kilohertz. So how did we learn all of this? Well, with large axons and, well, cell bodies are large enough, we are able to basically inject a current into the cell, and we can stimulate the cell to generate action potential. So it's an artificial process, but it allowed us to see some very interesting properties of action potential. So by injecting an electrical current, and this is the diagram of the electrical current being injected. So we have a period here that we have nothing, then we turned on the electrodes and we have a certain potential formed by our electrodes, which are represented here. And for practical purposes, these are nothing but glass tubes, very fine glass tubes filled with sodium or any other conductive liquid. So if you see this, if you can see this correlation between this injected action potential and this train of spikes, we have reached a certain threshold in depolarizing the cell, and the cell from that point on starts spontaneously generating action potentials. And I believe there is a slide there, but once we shut this down, this will shut down also. So the frequency of this, basically the time it takes for the next action potential to occur, it basically depends on this magnitude. So if the magnitude of the continuous depolarization is greater, the number of spikes per second is more. So as I stated already, the maximum firing rate that we have been able to observe is 1000 Hertz. Why? Well, it's because it is, for practical purposes, impossible to initiate another physiological action potential for about 1 millisecond after the previous action potential is finished. This period is called refractory period, and it coincides with what we saw previously already on the general diagram of an action potential. This 1 microsecond corresponds to the undershoot. And if you think about it, it makes perfect sense, because from an undershoot, it requires a lot greater depolarization to reach that threshold potential for an action potential to arise. So now, here is a clever little experiment that was done. With varying the magnitude of injected current. So in this first portion, we have a certain amount of depolarization of the membrane in a neuron, and obviously we do see some response. Yeah, well, we're injecting a current there, obviously, but nothing beyond that. There is no action potential, as you can see, no action potential. When we increase this current a little more, we have passed the threshold now, and we start generating action potentials. Great. Now we increased this potential more, and lo and behold, we started making action or generating action potentials that are much higher in frequency. So the membrane current and conductance, as you remember, current is the net movement of potassium across the membrane. It is facilitated by potassium channels, and the potassium channel numbers are proportional to electrical conductance. So the membrane potassium current is the driving force of an action potential. Now let's consider a following scenario based on a model neuron. What do we mean by model neuron? Basically a simplified neuron which has channels that are only of interest to us, which in this case would be the sodium and potassium channels. So what do we need? Well, first of all, we need the mechanism that will generate the chemical gradient of these ions. So we need sodium potassium pump that will separate the sodium and potassium on either side of the membrane. And obviously we would need potassium and sodium channels. So it is the pumps that establish the gradients and make the environment suited for neurons to be able to generate action potential. So for this particular example, we will assume that the concentration of potassium inside the cell is 20 times that of outside of the cell, and the sodium is 10 times that of or 10 times outside of the cell compared to that of inside the cell. So as we stated already, this is a model neuron, and in this case we only have sodium and potassium channels, sodium channel, potassium channel, and both of these channels at this given point are closed. If they're closed, even though we have the potential for potassium being minus 80 millivolts and for sodium being 62 millivolts, but because there is no open gate for any of the ions, there is no net flux, and therefore our voltmeter would read 0, no potential, right? Now, if we open up the sodium channels, the potassium channels, and we have stated already that we have 20 times more potassium outside of the cell than inside. So higher concentration of potassium inside the cell, and we open up, of course the potassium would want to leave the cell, and with each ion exiting the cell, it carries one positive charge with it, and over time, when it comes to an equilibrium, chemical equilibrium, we have generated a transmembrane potential with negative inside the cell. And in this case, because we have only the sodium channel, only the potassium channels open, our membrane potential would actually be equal to that of potassium, because that's the only thing at play here. Our resting potential is 65 because we have other ions that play a role here too. Okay, so we have our resting state. Now it is time to draw parallels between the stages of action potential and actually function of these channels. So at the resting potential, and remember this is our modeled neuron. So we have our resting at minus 80 millivolts, which is that of potassium, and we are at rest. So we have a dynamic equilibrium here. But then something interesting happens. The sodium channels open and sodium starts flowing into the cell, and concentration of sodium outside is much greater than that of sodium inside, right? So it starts flowing in, and it starts depolarizing the membrane because, well, the potential of sodium is 62 millivolts, right? And that's with respect to the cell membrane. So what happens? The action potential depolarizes. Then the inside of the cell becomes more positively charged. And then after that, the sodium channels close and potassium channels remain open. So the potassium channels start pumping their potassium out, carrying a positive charge with it, up to a point in which they come to, again, an equilibrium. And we have gone through this spike and came back to our resting potential. So what we're talking about is a result of years of work and some technical breakthrough technology, which is one of them is the voltage clamp. The voltage clamp right here. That was initially developed by Kenneth Cole, but it was extensively used by Hodgkins and Huxley. They actually received the Nobel Prize for their work in 1963. So punch clamp basically allows to fix the potential of a membrane at any given magnitude. And that allows certain experiments to be able to be conducted. So if we can set the voltage at a particular level, then we can see what's happening with, let's say, the flux of sodium ions, flux of potassium ions, and they were able to correlate the sodium and potassium as being the main players in an action potential. Obviously, their work was carried forward, and their initially described voltage-gated sodium channels have now been characterized on a protein level, on a genetic level. And now we know that these are transmembrane proteins, and they have basically a pore. They form a pore that is selective to a particular ion. But the protein itself is a single long polypeptide with 4 domains, and each domain consists of 6 transmembrane helices. So these are transmembrane helices. The interesting thing here is right here we have an array of amino acids that are charged. So what we have is we have our pore, we have a domain that is potentially can sense voltage, the electric potential. So, well, let's see how would that work. And the theory here is quite straightforward. Well, if we have a negative charge here, all these positive residues are going to be pulled towards the negative charge. right? And that keeps everything closed. Everything is nice and tight. But, and remember, action potential is an all or nothing event, right? There is no gray line there. If this action potential is destabilized, the membrane potential is destabilized, meaning the negative charge here is reduced. That allows these residues to move away because there is some bond strength that pulls these residues up. So if there is no very less counteracting force from a negative charge pulling them in, they will move up, and at this stage the gate is open. Now the pore here, the active the opening itself has a particular size, and let's say this is what the size of it is. And if you remember, an ion in a liquid solution is actually shielded with water molecule. It's hydrated shell. Anyways, the pore is such that it will fit a sodium channel which is hydrated, but it will be a very bad fit for a potassium channel, potassium ion being hydrated. As you can see in this illustration, it does not fit within the pore. Well, it is not to say that it completely retards the passage of potassium, but it significantly reduces the selectivity is several fold greater for sodium than for potassium, 20-30 times more. So therefore making a selective sodium channel. And in this particular case, a voltage gated sodium channel. So here is another rather elegant experiment conducted by Erwin Neher, in which a voltage gated sodium channel current was measured using patch clamp method. Patch clamp is basically, again, taking a glass electrode, patching a little area of the neuron and looking at what's happening in a current. So it turns out that we have something really interesting happening there. So here on this slide we have a representation of three different channels here, three different sodium channels. So when we are at minus 65 millivolts, everything is closed, nothing is happening, right? As we would expect. But when the membrane is depolarized to minus 40, something interesting starts happening. There are some variations from channel to channel, as you can see, in terms of time, duration, and whatnot. But one thing that is common between all of them is that once the channel opens and closes, it will not open anymore throughout this duration in which the potential remains constant. So this kind of closure of the sodium channel is known as inactivation. So that channel, that particular channel, will remain closed up until the potential drops and comes back on. So to look at what it looks like in a cartoon presentation here, our sodium channel is closed because Our membrane potential here is minus 65. Here in this case, it gets depolarized and the sodium channel is open, allowing sodium in. And then after a certain amount of time, it closes. Furthermore, it gets deactivated right here. Once we have this reset here, when it goes back to resting potential, then our sodium channel becomes ready for the next firing event. So there is a lot of fine tuning and synchronization there. The sodium channels, they open up with a little bit of a delay, which is very important, as you will see. They stay open for about 1 microsecond, and they get inactivated, which means that's it. They cannot open up until the next event occurs. And that causes what's known as absolute refractory period, when a number of sodium channels are deactivated to an extent in which no action potential can be generated after another. It requires the membrane potential to be reset. Here is another slide reiterating what we just spoke about. We have these 4 characterized events in which a resting potential, nothing is happening, the sodium channel is closed. Second, the sodium channel opens, then it gets deactivated, it can no longer open, and then it's reset and ready for the next firing event. So we generally learn a lot about the biology and physiology by studying disorders and toxins and such. For example, one such disorder, which is a genetic disease, I suppose, which is channelopathy. Channelopathy is basically a mutation in sodium channels that cause generalized seizures, epilepsy, basically. It could be as simple as a single amino acid mutation in a sodium channel. Besides that, we have some other tools to help us study nervous system, which is one of them is tetrodotoxin. Tetrodotoxin is one of the most potent toxins known, and it's generated by pufferfish, and apparently it's a delicacy in some cultures. Tetrodotoxin, basically, it can shut down sodium channels. It binds very, very tightly at a specific site outside of the channel, and it blocks all sodium-dependent action potentials. And therefore, obviously, it's obvious that it's fatal. Red tide, for example, is another common threat. The soxitoxin is another sodium channel blocking toxin. It is produced by dinoflagellates. It can be concentrated in food that we eat, like clams and mussels, shellfish, that they themselves feed on protozoans that produce the toxin. And occasionally, of course, this creates a red tide, which can be very fantastic when it's bioluminescent. But at this time, eating shellfish can be fatal, so stay away from it. The opposite to these toxins is also very detrimental. Obviously, closing the channel is bad, but keeping the channel open is equally bad. So the toxin, the batrachotoxin, which is released by certain frogs, they cause the channels to open at more negative potentials and to stay open for much longer than usual. As we have talked about this already, our channels open up, stay open transiently, and then they close. This toxin causes the channels to open up when they are not supposed to be open, and it makes them stay open longer than they should. And as a result, it scrambles with all of the information encoded within an action potential train. As you remember, the information encoded is by number and frequency of these action potentials. So if that potential is disrupted, the whole system is disrupted. The another one, the veratridine, and another toxin generated by buttercups. They also inactivate channels. But these can be used as tools for us to study certain channels if we know or certain physiological systems, not just channels. If we know what the action of a particular compound is, we can map, even on a protein level, how the protein looks like. If we know that a toxin binds to different sites in a protein, then we can use different toxins to infer what the 3D structure of a channel is. This is it for today. We will pick up the next lecture with voltage gated potassium channels. Thank you.